Questions from students?
So you guys mentioned that you measured the floral length rather than the inflorescence length. Can you kind of describe like what the difference between those two are?
So so the inflorescence length would be like one of those whole little, I don't know how to describe it, the whole bushel, the whole little part of the flowers. And then what we measured was this on the far left picture. This is one single tiny floret. I don't know how to describe that another way. Our measurements were very small because we were measuring the very small part, and the other group, theirs was larger because they were measuring the whole thing, if that makes sense.
Audrey, go back to your slide with the net on it. Yeah, use your pointer on your computer. We can see your pointer.
Oh, okay.
Yeah.
They were measuring this whole part.
Okay, yeah, that makes sense. Thank you.
Whenever you first collected data and your plants dried out, what did you do the second time to make them not dry out?
I can do that one. So we put them in ethanol, which preserved it, and it allowed us to go back and dissect it later, because the dissection process took quite a bit longer than we were anticipating, and so we really needed that buffer of preservation in order to be able to continue collecting over the course of multiple days.
Tessa, we're having some trouble hearing you.
Oh no.
That's all right. You're fine now.
Okay. Okay, I can repeat what I said. So the second time we put it in ethanol rather than just putting it in the fridge or leaving it out on the table, so that we could go back for several days to dissect and to count the pollen, because the dissection process took a lot longer than we were anticipating, because we just hadn't ever practiced it before. So ethanol is what worked for us.
Other questions for this group? Audrey, can you navigate back to the data slides? Yeah, was this the only data slide that we had? Is that right? Yeah, okay. And Angelina, you took us through this, right?
Yeah.
Okay. So for all of these, so for these two regressions, are the slopes of these regressions not significantly different from zero? Am I understanding you correctly there?
Yeah.
Okay, so these are basically not different from a flat line. Okay. So why would you expect patch density to have an influence on the ability of plants to pollinate be pollinated by a different goldenrod plant rather than the same goldenrod plant? Why were you expecting there to be a correlation there? Even though you didn't find one there in the end, why did you expect there to be one there?
Well, this kind of goes off of the chin diagram that I showed. If you have like a large amount of flowers, then it can attract more pollinators rather than just like one plant standing on its own. The like bright yellow field attracts the eye of different pollinators like bees and wasps.
Okay, when you guys were doing field work, did you see a lot of pollinators out there?
No, because it was starting to get a little cold. And I don't know how we managed to do this, but even though it was the end of the pollinator season, our the data that we collected was young, and so it was kind of like a few caveats to our results because there weren't any pollinators really out to pollinate the stigmas, and the stigmas weren't really open all the way yet.
Okay, so I don't know that you guys made this terribly clear, especially for the other groups that weren't actually as deeply ingrained in your project. Explain to me kind of how the stigma and the anthers relate to one another as the flower develops, and how that enables you to look at different parts of the stigma and determine what pollen on that stigma are stigma from that individual flower versus pollen from another flower.
So the way that the stigma grows out of the flower is, you can see it goes up through the flower. And so while the stigma is growing up, all of the pollen from the flower and from the anthers is rubbing off on the outer edge of the stigma. And then when it opens up, that's when the other stigma from other flowers falls on the inner part.
Okay, good. Go back to your data file, or your data slides. So on this data slide, where are your data and where are the Thursday Labs data? Do you know where the breakpoint is for that? Do you guys remember which patches were yours versus which patches were the Thursday Labs data? I myself can only remember it approximately.
Okay, I think ours was the like first half of most of the data sets. But when they got combined, it got a little hard to decipher.
Yeah, so your data set is the first 16. So most of the data in this data set are from the Tuesday lab. What do you note about the difference between your data and the data from the Thursday lab? By patch. So your patch is patch 1 through 16. Their patches are patches 17, 18, and 19. I'm looking at the upper left and lower right figures.
Our measurements were much larger. We had larger ratios and a larger number of pollen grains found.
You did have a lot of pollen relative to those, and yours was much more variable than the other one. And this probably has to do with the fact that you were actually measuring a variety of different stages of maturity of the stigmas. So I hadn't thought about this when I was running the data, but those last three patches are the only patches from the Thursday lab. They're all uniformly low and really not very variable. As opposed to your guys, you have a lot more pollen, and the pollen counts from stigma to stigma are very variable. And so that also probably speaks to differences in methodology between the two groups.
We also, since we didn't collect at Martha Feet, we collected off the side of a pretty busy road. So we're not sure if the traffic that was going through that road had any effect on the goldenrods that were next to it.
Sure. Yep. Any other questions for this group? All right, let's thank them and we'll move on to the next group. And the third group is going to be the pull and load group. All right. Take it away.
All right, can you guys hear me?
Yep.
Cool. All right, so our group consisted of myself, Sophia, and Alexis, and we looked into the pollen load capabilities of a couple of different types of insects. So I just wanted to put the image on the right into the presentation because we saw a lot of this over the course of collecting our data and analyzing it. This is a hemoseptometer. And so I wanted to give you guys like an idea of what we were looking at while we were analyzing the data that we got. So it'll come up more later. All right, first we'd like to acknowledge Dr. Klinsky and Dr. Judd for their contributions to this project, providing us a space to work and giving us much needed direction. We'd also like to credit Alex Epp, who was in our group for a short time, but and helped us with the collection process, but sadly left in the middle. We'd also like to thank the Martha Laffitte Thompson Nature Sanctuary. So insects, goldenrod, and location. Our primary location for the study was the Martha Laffitte Thompson Nature Sanctuary. This location has a consistent population of Solidago canadensis, also known as Canada goldenrod. This location is also isolated from day-to-day human activities, so we didn't really have to worry about anyone mowing or really messing with the goldenrod population there. We also used a secondary location for the insect collection process because there was a better bloom of goldenrod outside of the sanctuary. So similar to the last group, we went out by South Liberty Parkway to collect insects. So some key background information about goldenrod. This information is based on our own observations and observations by Blackwell and Powell in 1987. Its pollen has been observed to be relatively sticky and can be found all over the body of wasps, including the antenna. Solidago canadensis also stays in bloom relatively late in the year, and it has been observed to be a source of nectar for insects all the way through late October. Obviously, this is weather dependent, and we saw this as a problem in our own experiment, but it can be in bloom pretty late in the year. So moving on to insects, after visiting the nature sanctuary, we observed 4 different insects having a strong presence of goldenrod among patches. These observations would become the basis for our study, and the most prevalent insect from an observational standpoint was the goldenrod soldier beetle. And then we also noticed a lot of activity from paper wasps, honeybees, and bumblebees. So some important things to take note of here are the size differences. So the paper wasps and the bumblebees are clearly the biggest bugs. The other two are pretty close in size, them being the honeybee and the goldenrod soldier beetle. Unlike the other 3 insect species, goldenrod soldier beetles do not live in colonies. Paper wasps live in small colonies, and obviously the 2 bee types live in large colonies. So what is pollen load? Pollen load is a measure of the amount of pollen an insect can carry on its body. This only includes the pollen that can be used by other plants in the process of pollination. So in the image to the left, we see a bee species with a large pollen basket, which will eventually be used to feed larvae in their colony. But we obviously can't include this pollen basket in the count of measuring pollen because the pollen is so tightly packed on their legs that it becomes useless for plants. And so in their process, how do we analyze pollen load? In a study done by Guyan and Waddell in 1987, pollen load was measured by first catching and killing the insect, removing the pollen from the insect's body by vortexing, centrifuging this vortex mixture, adding stain to this mixture to differentiate between pollen types, and then pipetting this mixture into a hemoseptometer and counting individual pollen grains. And so this ended up looking like the first image on our slide. So we pipetted this mixture onto our hemoseptometer and looked at basically this onto under a microscope, and we counted individual pollen grains, and that's how we determined pollen load. So if we go back, and then the last thing on there is removing the corbicula. That's basically just this specific study cut off the pollen basket before actually analyzing the amount of pollen load, just so they could get a more accurate number in terms of the amount of pollen an insect can, the amount of pollen an insect can spread. Okay, so our guiding question for this presentation or this study was how do patch density and pollinator load affect the pollination efficiency of forward insects on Solidago canadensis? And then I already talked about pollen load, but patch density is the ratio of patch size and the number of goldenrod counted in each patch. And our hypotheses, we split them up into 4 different parts for this study. First is that goldenrod beetles would be the most specific pollinators to goldenrod and the most abundant on patches. Second, different species would carry different amounts of pollen, with the bumblebees carrying the most. Bigger patches of goldenrod would have more pollinators. And finally, patches further from the edge would attract more pollinators. So I'll hand it over to Sophia.
So to test our hypotheses, we went on to the Martha Lafitte Sanctuary and we used the wandering quarter method, which is the image you see on the bottom right of the screen, which was basically we created 90-degree angles with our hands and then we picked patches within that angle so that we didn't pick the same patches to count. So within those patches, we picked individual goldenrod plants and we counted any insects that we saw on that on the rods. But obviously, like Joseph said, we didn't have much luck at the Martha Lafitte. So we had to go to the spot on the side of the road that Dr. Klinsky directed us to. And that's where we saw most of the insects because the goldenrod there was still blooming. And so we measured and captured the insects off any of the goldenrod strands that we measured initially, and then to take them back to the lab, we had to catch them with nets and place them in kill jars, which was just a mason jar filled with ethyl acetate. And then once we went back to the lab, they were placed in vials of ethanol where they were vortexed so that we could separate the pollen from the body, because the body would obviously have an effect on the pollen load. So we wanted to remove the body. And then after we removed the body, we centrifuged them to separate the pollen from the ethanol so that we could analyze it. So like the first slide, there was a hemoseptometer, and we placed the pollen as well as the ethanol onto that slide and put it under the microscope so that we could count the pollen grains within those wells.
All right, so this is what we got for our original data. And so you can see that the 4 different species that we looked at are on the x-axis there. And then on the y-axis is the load. And so that's basically just how much pollen we counted on those hemoseptometers. And then we multiplied it to get this big number. And basically it was just extrapolating by like how much would actually be on the entire insect. So the biggest, oh, can you go back to that last one? Sorry. So on this slide, the most important thing to point out is that Bombus, and that's the bumblebee species, and there's just that one extreme outlier that I colored in blue there. And so you can tell that like there isn't actually that much like up there, and that's really the only outlier that really has that much pollen on it. So then on that next slide. So our goal with that data was to run an ANOVA test, and to do that, our data had to be normally distributed, and it had to not have very much different or variation between the different species. So this is a QQ plot that we use to look at the normality of our data. And so this is really similar to what we looked at in the Galapagos finch case study. So you can kind of see that that data is pretty much a straight line along with that diagonal line there, except for that one Bombus outlier that has just that ton of pollen on it. So then in the next slide, you can see that we also looked at variation, and we did that by using the Levine's test. And you can see that the variation isn't that much different between the different species, except for that Bombus has a ton of variation around it. So then to be able to actually run an ANOVA analysis on this, we needed to take out that Bombus outlier so that everything would be like it would fit the assumptions of the ANOVA. So this was the plot that we got for the QQ plot after we took out that Bombus outlier. And you can see that once again, it's mostly along that diagonal. Now that we took out the Bombus, it fits it pretty well. Then the next slide shows our new variation. And now the biggest variation is around that wasp. And there isn't very much variation across the different species. So then we did an ANOVA test on this. Oh, last slide, sorry. Yeah, so we did the ANOVA test on this, and we found that we had a P that was actually smaller than the alpha, so at least one of those was different. And we had to use the Tukey's model of significant difference to find out what the actual difference was there. And we found out that the only difference was that the wasps were different from the goldenrod. And then now on that next slide, we also did a regression to see if the pollen load was affected by the patch area. And because we only had 2 patches from the side of the road that we could actually count insects on, you can tell that that's pretty much a completely straight line. So no, our regression did not show that pollen load was affected by patch area.
Okay, so just to reiterate, Alexis, a little bit, the results of our analysis were that the only significant difference in pollinator load was between paper wasps and goldenrod beetles. And so this difference could be attributed to some morphological differences between the 2 species. For one, the wasps are on average larger than the soldier beetles, and the wasps have been observed in the past to visit plants more frequently, given on a given day. That's from Blackwell and Powell again. So the fact that one thing we need to point out is the fact that there is a small difference between 2 different insect species. This kind of indicates that we would need more data, and that more data might show that there would be other differences between other different pollinator species. And lastly, we also found no significance between patch area and pollinator load.
So a couple restrictions we had that altered our data as well as changed our procedure a little bit was it was really late in the season, so there weren't a lot of blooming goldenrod. So we didn't find a lot of insects at the Martha Lafitte Sanctuary. That's why we had to go on the side of the road. And then we only considered goldenrod pollen, meaning when we analyzed it, we didn't separate what type of plant the pollen was from. So while we did catch the insects on goldenrod, they could have contained other pollen from other plants, but we did not differentiate that. And then future studies, so a couple that would just, if we did this again, we'd probably better our experiment. If we obviously chose better timing to where there's lots of blooming goldenrod. And then, like I said, we needed to look at the types of pollen rather than just look at pollen as a whole. And then another study that we were kind of interested in is taking the same species and counting the pollen load on them, but collecting them from different locations to see if the different locations could have an effect on how much pollen that they carried. And then this is our works cited page.
Questions from students?
Yeah, I had a question. Like throughout all your research and stuff, is there something that you would do different, to make it easier?
Yeah, I think one thing that we could have done to make this process easier was at the beginning, learn more about what pollinator load is. So before we actually went out into the field, I wish we would have done a little bit more background research into, how insects carry pollen, just so we have a better idea of what we're working with before we just kind of jump into it. So, if anybody else has any comments.
So you talked a little bit about how like the morphology of the insect might affect how much pollen they collect, and I was wondering if you could maybe elaborate that on a bit more. Is it like the anatomy of insects, or like what exactly do the insects do in order to be more successful at collecting pollen?
So that was kind of based on some observations that we made. So we observed, and based on some data compiled by the Missouri Department of Conservation, the wasps were paper wasps were on average much larger than the goldenrod soldier beetles. So that can kind of indicate that while both of them are fairly hairless, and another thing that I could point out is that hair usually has a correlation with the ability to hold pollen. So hairier insects can on average hold more pollen because the pollen sticks to the hair. So basically what I was saying with that was that because the wasps are larger, they have more surface area for pollen to stick to them. And so that is one thing, one morphological difference, just the size. And then the other thing that I said was, um, oh yeah, wasps move. So in a past, not a past study, but it was just like an observational piece that I read by Blackwell and Powell in 1987. They observed wasps moving from goldenrod to goldenrod up to 11 different goldenrod plants in a single day. Um, so just when we're looking at that data, we can see that the wasps seem to be more mobile than the soldier beetles, just because they're larger and because they're collecting pollen they're collecting resources for their actual colony, whereas the goldenrod beetle is kind of just sustaining itself. So those are just assumptions that I made um, based on our data and based on past observations.
And we kind of saw that with the outlier that we had. We had one bee that was just huge. So obviously you saw on the graph, it kind of threw off our data, which is why we got rid of it. Because it just created right here, it created a huge outlier because it was so big and had so much.
Was there one species that was more difficult to catch than another?
The wasps were pretty scary because they're fast and they're strong and they have big stingers. So that was probably the most intimidating to catch. The soldier beetles were pretty simple. That you could literally, at first it was kind of hard to catch them because we didn't know what we were doing. But after we got the hang of it a little bit, you kind of could just pick them off of the goldenrod and put them into the kill jars. Um,
Another thing with the goldenrod beetle is like there was just so much on every one of those inflorescences that you could just kind of swing the net at the inflorescences and you could pretty much guarantee that you'd get 2 or 3 of the goldenrod beetles in there.
Yeah, that's true. And we also had, whenever we were catching I actually remember catching the bee that became our outlier. It was so big that it took like 2 or 3 minutes to actually die in the kill jar, and it was buzzing very loudly in the jar the entire time. So yeah, and it barely even, we had to take the insects from the actual vials we used in the field, and we transferred them into different little not jars, but glass vials, so we could separate the pollen that we collected from the insect. And so when we put it in there, that barely even fit in the actual holding glass. So yeah, it was huge. It was a big bee.
Other questions from students? Okay, so you guys kind of finished off from the question from Michaela, talking about size. You ended up not going back and measuring size, mainly because we kind of ran out of time and then everybody dispersed. Do you think size is as important, or do you think it's aspects of the morphology of the different insects that affect their ability to carry pollen?
I think it's a little bit of both. I mean, the biggest thing about that bee was obviously it was bigger, but it was also hairier. So the fact that it has more hair means it can hold more pollen. Um, but if we think about it more, um, there's a bigger population of the smaller honeybees visiting the flowers. So does the actual size of one bee outweigh the effects of like 5 honeybees? Probably not, but I mean, obviously, with our results, the size of that bumblebee did have some impact.
So how many of those bumblebees did you have?
We only had we had one that was really big, and then we had a good, about probably a handful that were not like the size. They were about around the size that they're around this normal 1.5 to an inch size. The one that we were looking at was probably, it was much thicker. It was probably still like 1.2 inches, but it was the size of the tip of my thumb probably.
So even though goldenrod soldier beetles were probably the most abundant insect out there, how would you rate them in terms of their ability to serve as a pollinator for goldenrod?
Yeah, so they were not carrying very much pollen is what we found. And I guess it didn't really surprise us because they're not very big. They don't have very much hair, so we weren't expecting them to carry very much pollen. But as you can see, like it's really not very much at all. Like they had the lowest mean there. So they probably were not the most efficient for carrying the pollen.
So when you think back to some of the other presentations that you have seen today, especially the life history presentation, what do you think the role of the insects are in terms of pollination efficiency for these goldenrod plants, given that what we learned from the life history group is that they're pretty good at getting seed set in terms of number of viable seeds up, but they're still, 65 to 90% is the range of viable seeds in terms of percentage of total seeds produced. I guess what's the question I'm trying to ask? Based on the life history data combined with what you found with your insects, do you think any of these pollinators are particularly efficient? And what would you maybe like to know more about if you were going to make a better determination of that?
I guess I'm still a little bit confused on the question.
So if you go back to the life history data set, they found that in terms of the total number of seeds that the plant produces, there's only a proportion of those that are actually viable, that have been fertilized, been pollinated and then fertilized. So there's a big pool of seeds, but some of those are not viable. And those not viable ones are generally ones that were produced but weren't fertilized. And that percent viability, the percent of the total proportion that is viable, ranges from, according to their data set, about 65% to about 90%. So as much as 35% of the seeds that a inflorescence produces aren't viable. And so my question, given their data set and given your data set, what do you think in terms of, is this suite of pollinators very efficient based on what you know about pollen load and what they learned about seed set?
So I would say that since most of those seeds, or some of those seeds, are not getting the pollination that they need, that no, these pollinators aren't as efficient as the goldenrod might need. And it might be interesting to see if there was a species of pollinator for the goldenrod that maybe carried a little bit more and was a little bit more specific to goldenrod, because goldenrod is kind of unique in that it needs pollen from other plants rather than itself. So it's really important that those pollinators are going back and forth to different goldenrod plants and not distributing pollen on the goldenrod that is like useless. And most of our species here are like general in that they go to a lot of different flowers and don't just specify on the goldenrod. And the goldenrod beetle, they do mostly specify to yellow flowers, and around here that's pretty much the goldenrod. But since they don't carry a lot of pollen, like that's not really helping out the goldenrod a whole lot.
Yeah, and I'll add on to that by saying that while we were actually looking at while we were counting pollen grains on under the microscope, we really didn't see that much. So if we tack that on with what Alexis is saying, we can, and what we know about the life history group from the life history group. So we have only a 35% viability. Plus, we didn't really see that much pollen on these insects anyway. So yeah, no, I would also agree with Alexis and say that this suite is not that efficient as a group of pollinators for goldenrod.
So Alexis just mentioned that these pollinators are all, for the most part, generalist pollinators. They're visiting lots of different kinds of flowers. Did you see any other pollen? Go back to your slide that has the picture of pollen. Oh, no, that's not your slide. That was the slide from the pollen limitation group. So goldenrod pollen is what we call echinate pollen. It's this little sphere with little spikes coming off of it. Did you guys see any non-echinate pollen in your samples? Pollen that looked morphologically different than the goldenrod pollen?
So we just counted all types of pollen for our study. When I think about it, I don't remember seeing anything other than grains that were circular and had spikes sticking out of them. But there very well could have been. We just counted them all together because.
And to kind of add on to that, whenever I was counting that, because I was the one that counted the huge bumblebee, and it had like 27 grains of pollen just within that area. And there were a couple of them that I do remember having a different shape, like a little bit more like a bean rather than like that circular sphere.
Okay, so, and that is, bumblebees are very much a generalist pollinator. So one of the things that you might do in the future is distinguish between echinate pollen and non-echinate pollen in your counts, of course. Any other questions from students? All right, let's thank this group. And this leads us to the last group, which is going to think about reproductive success of goldenrod, but from a slightly different angle, looking at flower galls. So the flower gall group is the last group for today.
Can you guys hear me? Just to make sure.
Yeah, hold on just a minute.
Okay, sorry.
I'm ready whenever you are.
All right, can you hear me?
Yep.
Okay, so our group consisted of myself, Sydney, and Kat, and we looked at the effects of galls on goldenrods. I would first like to acknowledge the William Jewell Biology Department for giving us sufficient resources to complete our studies, Professor Klawinski and Professor Judd for their guidance and assistance throughout our research, the Martha Lafitte Thompson Nature Sanctuary for letting us do research on their land and also use resources from it to complete our studies, and the other Gall group with providing us with comparable data. Here we go. Pollen from one flower drops or is rubbed on a pollinator, and is then carried to another flower. And then the next flower is then supposed to use this pollen to produce seeds. These seeds are usually carried to other locations through wind, water, or insects. Goldenrod flowers can be distinguished by their long wood-like stems with bright yellow inflorescences at the top. Goldenrods tend to live mostly in moist locations, forests, fields, roadsides, and compost fields. In looking for other studies, the number of seeds produced was not listed, so we determined that it's anywhere from around 400 to 18,000 seeds. Most of these seeds are then dispersed, usually through wind. However, some of these seeds are affected by galls. Galls look like funny or fuzzy domes throughout the top of the inflorescence of the flower. They are formed when an insect lands and lays their eggs here. The saliva released from the egg triggers a response in the plant to stimulate growth in this area, thus forming these mutated galls, which you see on the right, which the eggs use as shelter and a source of nutrients. These galls are usually located throughout the inflorescence of the plant. Given this location, they are believed to affect the seed size of the plant. Our questions were, what effect do flower galls have on reproductive success in goldenrods? And do gall-infected plants shift investment from gall-infected flowers to healthy flowers in ways to affect seed size? Our hypothesis was the more galls that are present, the lower the seed set, because the flower would allocate energy to the galls and not seed production.
So we conducted our research at the Martha Lafayette Thompson Nature Sanctuary. We had 3 separate transects that we collected goldenrods on. The red transect was our first one. Then you have transect 2 is the blue line. And then transect 3 is the yellow line. We set out with the goal to collect 30 plants from each transect measuring, and then 60 total. We needed to find 15 of those plants to have galls. However, we didn't find enough goldenrods on these 3 transects, so we ended up having to kind of go off the paths and search for them. Next. In the field, we measured the standing height of the plant, or just the height of the plant that it was standing at when you approached it. And then we measured max height. We stood it up to a 90-degree angle from the ground. And then we measured the length of the inflorescence. So the inflorescence starts about where this red line is on the photograph. It's that lowest branch. We also measured the size of each patch that these goldenrods were in, and then the density of the patch. Hold on, Connor, go back. So as we came across a galled plant, we cut it at the base of the inflorescence after taking the appropriate measurements, placed it in a labeled paper bag, and stapled the bag shut. In lab, we measured the weight of each inflorescence. Okay, now next. We removed all the seeds from each of the 15 collected inflorescence, and then from each, we selected 100 seeds and evaluated them for their viability. You determine viability of a seed based on if it's round and plump, then that's a viable seed, and if it's wrinkled, it's not. Then we weighed each of those 100 seeds and divided that number by 100 to get the seed size. Then all 100 seeds from each plant, so 1,500 total seeds, were placed in a graduated cylinder to find the volume of 1,500 seeds. And then we did the same for all the seeds from one plant, placed them in a graduated cylinder, and found the volume. Okay, next. And then we use these equations to calculate the estimated number of seeds and then the seed set. Next. Sydney.
Okay, so for our results, so we kind of started with looking at the relationship between plant height and the seed set that we found. So for plant height and total seeds, like on the plant overall, we found that as like a plant is taller and the height increases, so does the total amount of seeds. And then that's shown on like the graph on the left. And then on the graph on the right, it kind of shows that like the plant height, as plant gets taller, the seed set is also going to increase, like as the total number of seeds increases too. Because as Kat just said, like to find the seed set, you multiply the total number of seeds times the proportion that are viable. So that kind of like shows that relationship, how the plant gets taller and there are more seeds total. So then there's a higher seed set. And then you can go to the next slide. So next we looked at the relationship between inflorescence length and plant height. And we found that there's like no significant relationship between these two. So that led us to our next slide to look at the individual relationship between like inflorescence and seed set. And so we found that on the left, you can see that there's no relationship or no significant relationship between the inflorescence length and the plant having a larger seed set, but inflorescence mass did affect it. So when like the inflorescence has a larger mass, then there's going to be like more total seeds that are causing that mass to be bigger, and that kind of explains like the higher seed set that we found. And then you can go to the next slide. So to look at like the galls effect on seed set, we looked at how the inflorescence length affects the number of galled branches on the plant, and we found that the larger inflorescences don't necessarily have like more galled branches, so there's no significant relationship there. But you can go to the next slide. We found that galls did affect the seed set in the plant. So as there are more galled branches on the plant, there's going to be a higher seed set. So this kind of like rejected our hypothesis that when there are galls on the plant, there's going to be less viable seeds, because we thought that it would allocate energy towards like the gall because it kind of puts nutrients in that, and that would take away energy from producing seeds. But as you can see in this graph, when there are more galled branches, it actually results in a higher seed set. And you can go to the next slide. So compared to non-galled branches or non-galled plants, we even found that goldenrods with galls are going to have a higher seed set at some masses. So again, that kind of like rejects our hypothesis, because the seed set is able to like increase even in the presence of galls.
Questions?
I have a question. So can you guys describe like your selection or your collection process? Like, what did you actually get off of the goldenrod, and then like how did you turn that into data for these graphs?
So we probably should have learned a lot more about galls before we actually went out. And there's so many different types of galls, so we probably should have learned more about, specifically about the goldenrod flower gall. But when we went out in the field, let me just go back to right here. So we were looking for these on like the flower of the goldenrod, and there wasn't a lot out there. So like Kat said, we didn't find 15 in our transects, so we had to go to random places and find them wherever we could.
Joseph, did that adequately answer your question?
Yeah.
Members of the life history group should have a number of questions, I would suspect. Are you guys just tired?
Yes.
Okay, keep it up for just a little while longer.
I guess I can ask, what was the similarities and differences between the experiment that you guys did and the ones that you had researched?
So the the problem with that was that there is little to none like research on this stuff. So we couldn't really find any studies about galls and affecting the seed set, whether it increased or decreased. So I can't really answer that question for you. I'm sorry.
Yeah, I think we found a lot on like the gall insects and like the bugs that kind of form the galls on the plant and how like they affect the goldenrods, but not necessarily like galls on seed set.
And a lot of the galling literature out there is on leaf galls and stem galls. They're right. There's practically nothing out there about flower galling. Other questions from students? Sydney, you were the one covering the results. Can you go back to the slide on looking at inflorescence mass? Well, let's see. Oh, let's see. Yeah, inflorescence mass versus seed set. This is your guys' figure. Go to the last figure of the presentation. Okay, so where did the red data come from?
Well, we collected both galled and non-galled, so we measured the inflorescence mass of non-galled goldenrods also. So.
You didn't collect non-galled plants. You collected only galled plants.
We collected some of both. It was like 15. We got like 15 galled plants, but like 90 plants total.
Okay, but you didn't bring any of those plants into the lab and take seeds from them, correct?
Yeah, correct. I think to answer your question, aren't those orangey red data points from the life history group?
Oh, they're definitely from the life history group. So here you have two relationships. In the life history data set, there's no significant relationship between seed set and inflorescence mass, but in your data set, there is a significant relationship between seed set and inflorescence mass. And even if you remove that one outlier that is on the far right, the upper right blue dot, that's not going to change the slope of that relationship at all. It's right in line with all of the other data. So Sydney, I think you gave us a slightly incorrect conclusion on this slide. Can you repeat your interpretation of this slide?
So we said that like when galls are present on the goldenrod, there's like a higher seed set, especially at higher masses than ungalled plants.
So somebody else take a shot at this. How would you interpret this figure? And this is possibly my fault because I didn't probably spend enough time going through the analysis that generated this particular graph. But this is done with an analysis of covariance. It basically looks for differences between, in this case, non-galled plants and galled plants after statistically removing inflorescence mass. And so in the same way that we could use residuals to remove inflorescence mass, we can also do a thing called the analysis of covariance. And what that analysis showed, it's the last analysis in your R-scope, so go back and revisit that as you are writing up your paper. What it does is it basically says if all the plants had an inflorescence mass, say of 2,000, if 2,000 was the average inflorescence size, it basically takes each of these points that are the blue points and moves them down to 2,000 if they're above 2,000, but moves them down parallel to the line, and then moves the ones below 2,000 up to 2,000 parallel to the line. Whoa, and I just lost my screen. Oh, it's back, okay. And then makes a little cluster of them at 2,000, but as a result of moving these things down the slope of the bluish line. And it does the same thing with the red ones. It takes the red ones and says, well, where would all these points be if they weren't spread all across inflorescence mass, but if they were clustered right there at 2,000? And what you get out of that is you get a cluster of points clustered around the red line at 2,000 and a cluster of points around the blue line at 2,000. And in this case, the red cluster would be higher than the blue cluster. And when you go and look at the results of your analysis of covariance, you'll find that there is a difference between galled and ungalled plants. And it's a difference such that ungalled plants have more seeds than galled plants. And so in this case, galled plants would have a lower seed set than ungalled plants. And so what does that tell you about the effect of galling on reproductive output in this species?
That, yeah. Galls decrease the seed.
Repeat, please.
That galls would decrease like the seed set.
Yeah, and that's kind of exactly what you would expect, right?
Correct.
Yeah.
So those of you in the life history data set, what's different about their relationship between seed set and inflorescence mass and your data set on seed set and inflorescence mass? Now I'm asking a question of another group.
Oh, I can answer. The slope of their data is steeper, like closer to 1 than ours.
Yeah, and yours was not significant and theirs is. And part of it is that you had relatively few data points, but it's also a much shallower slope, which I was kind of surprised when I saw the relationship between inflorescence mass and seed set of your data set because there wasn't a relationship. And then I was super surprised when I saw the gold group, where inflorescence mass was highly correlated with seed set, because those would be exactly the opposite two results that I would expect, because as an inflorescence has more and more galls, it should reduce the seed set such that the inflorescence mass should become decoupled from seed set the more galls you get in your inflorescence in the gold plants. So this figure here is a little bit perplexing to me, and that I think I need a lot more data to try and get a handle on what the true relationship might be. But right now, it looks like it's consistent with the idea that the galling reduces seed set. So make sure that the three of you guys have that clear in your mind as you go to write this up. And that speaks to one of your hypotheses at the beginning. In your conclusions, you didn't really talk much about what you might do differently. What kinds of things might you do that might improve your insight into the effect of galling on reproductive output in the species?
Well, I think as you were saying, we definitely need more data.
Sure, and you have a good amount of it, but more data is always better. But that's not the. It doesn't require much creativity to think, oh, more data would be better. Think a little more creatively.
So we didn't have a lot of the measurements.
Sorry, Sydney, I can't hear you.
Oh, wait, can you hear me now?
Yep.
Okay. So we didn't have a lot of the data for like patch size and stuff like that, like relating to the patch we are in. So we talked about like maybe if we did that and kind of examine the relationship more between like the patches and like the plants in the patch and what like amount of galls they have.
Okay. Why would the patch size influence galling potentially?
I know we talked about possibly like taller plants in a patch are more likely to have galls because they're up above the like line of the other goldenrods.
Okay, so there might be some effect of just attracting insects in general that attracts more gold insects because you have more plants and things like that. Who's controlling the PowerPoint?
Connor.
Go back one slide. Go back another slide. Another slide. Okay, go forward 2 slides. So this is looking at seed set and the ratio of gold inflorescences to gold branches to non-gold branches. So you just counted the number of branches in an inflorescence that had galls on them. Is there maybe a better way of going about kind of quantifying what's the degree of galling in a particular plant? Because this also surprised me. The more galls you have, the more galled branches you have, the higher your seed set is, which also is not what you should expect to see. Well,
maybe
what would be maybe a better way of doing this?
Maybe counting the seeds of each individual branch and seeing if, you know, a galled branch had less seeds than a non-galled branch.
Okay, but that would be a pain in the butt, separating all those branches and getting the seeds off of each individual branch of the inflorescence. Is there something that is maybe more informative but easier to accomplish? What was your criterion for determining if a branch of an inflorescence was a gold branch or not?
It had galls on it.
Yeah, 1 gall or 2 galls or 20 galls?
Didn't matter.
Didn't matter. So what might be a better way of kind of measuring degree of goldness?
Counting individual galls and not one branch.
Yeah, and Dr. Judd and I are kicking ourselves for not having you do that instead of what we had you do. because I think that would have possibly given you a different result where this figure is concerned. So this is the nature of anything that you do the first time. The first time you do it, you see all the ways in which you could have done it better. Any other questions from students? All right, let's thank this group. Okay. Heart. Yay. Okay. So usually when we're doing this in a room together, we generally kind of have a little debrief sort of about what your experience in lab has been like. But your experience in lab this year is very different from the experience of lab in previous years, because the structure of lab had to be kind of fundamentally altered. So I do want to have a little debrief with you, but it's going to be somewhat different. Here you are, last day of lab. You've done a presentation. All of your presentations were surprisingly good, given the circumstances under which you've been asked to function this year. I'm actually quite pleased with them. How are you feeling here at the end of lab?
Stressed out.
Stressed out. Okay. In that way, you're like every first year class that we've ever had who have gone through this process of doing research for the first time. What else are you feeling here at the end?
Honestly, I'm just really surprised it all came together, because we built presentations on Zoom, something we've never done before.
Yeah, so the act of how you put this together is different when you're having to collaborate on Zoom rather than if you can just sit down in a room together. That's I think a much harder task. What else?
I'm just glad that you and Dr. Judd helped us a lot during the process, because I don't think we could, we could write our method and hypothesis and questions for the introduction, and like all you basically prepared for everything.
Well, that's kind of our job when you think about it. If we're not doing that, we're really falling down on the job somewhat. Anyone else? At the beginning of the semester, when we kind of outlined what we were going to be doing with you, what were your thoughts?
I was scared I wasn't gonna be able to do any of it.
Oh, why is that, Connor?
Because first I didn't really know what a goldenrod was, and I had no idea how I was gonna collect all this data.
Okay. But what got you through it?
Lots of research, and then actually going out in the field, and I guess collecting the data myself and realizing, this isn't, horrible.
It isn't horrible. Oh, that's one low bar to get over. Anyone else?
I was kind of worried about how like team dependent it was going to be, and I was afraid that like if I messed something up, how much it would affect like other people or vice versa. But we were able to communicate well enough, and even though we did have errors in our lab, like we were still able to communicate that, and I think have like for a first go a pretty effective research project.
Yeah, errors are always common the first time you're approaching research. And I mean, you even see that Dr. Judd and I, who we tried to do our best to kind of think through what we wanted you to be doing, we ourselves would go back and change things. As a matter of fact, one of the things on our list of things to do over the Christmas break is to go through and kind of rework each of these projects to take what we've learned from you guys going through this to, for example, improve the protocols, make protocols clearer, alter what some of the data are that the groups are doing. And so even we learned a lot from doing this as a result of this being our first foray into thinking about goldenrods. Other reflections on kind of how you felt when you first embarked on this whole thing?
I was really kind of excited to be able to spend a semester just like kind of doing our own research. Like previously, anytime we've done anything in a lab, it's just been like, here are your set of things that you do in this 45-minute period, and then you move on.
Normally, what you would do in this class is you would have generated your own project, and some students find that even more daunting than generating a project that was kind of developed for them and handed off to them. But yeah, this is different in that you're spending a long period of time thinking about this thing, and hopefully that's useful and beneficial to you as you move through the program and anticipate doing undergraduate research as a junior or senior. Other thoughts?
For my lab group, at least, we definitely got a lot closer as the lab went on, and I think that helped us as we were preparing for the presentation and doing our research, because we met so much outside of lab time to go to lab and look at our stigmas. And so it was nice knowing that we had people who supported us and people that if we did have questions or got confused, we could ask our own lab members too.
Okay, cool. Other reflections?
This is more of an anecdote, but this project reminds me of like the science fairs that I had done in like high school and even middle school, except like really condensed down time-wise. And I feel like approaching it that way kind of made me more excited about it, even though it wasn't a topic that I probably would have chosen for myself.
Mm-hmm. So in science fairs, how long do you have to work on those?
We had like all year.
Okay,
So like
yeah.
the school year to the May.
Did the compressing it into a semester make it feel a little more stressful?
Yeah, it was definitely more stressful than what I had experienced, especially since like we didn't get to create our own deadlines. But
Mm-hmm.
I don't know. I feel like in the end, like if it was interesting research, if you actually like changed your mindset into something like, oh, I actually want to know what the results of this experiment are.
You never thought you would want to know anything about goldenrods, probably.
Well, you know, sometimes like I feel like we just go through the motions of like, okay, now I got to do this. But whenever you're actually invested into the like scientific process, it makes it a lot more fun and a lot more interesting to go to class and to do lab.
Okay. Other reflections? Sophia and Joseph, you just turned on your camera for some reason.
We felt bad because no one else has their camera on.
I'm the only one here with my face on screen. Thanks for that pity camera nod. Did you ever at any point think to yourself, oh my gosh, I'm never going to get this done?
Yesterday afternoon.
Yesterday afternoon. Okay. So as you think about the process, it's a difficult process. It's a time-consuming process. At times you think that you can't get it done. Having done it and gotten it done, you guys did a fine job on your presentation. As a matter of fact, Joseph, you had a very nice introduction in terms of kind of setting up and giving the background information that I found useful, and I think everybody else found useful as well. How do you feel about going forward? Like, does this make you think, oh, I'm never going to take another biology class again? Or does it make you think, oh, I can probably crack this biology nut?
One thing I'll say is I think it's easier for me now to interpret and find good literature to use, especially for background. I kind of have a better filter now, I feel like, after having to do it. I'm not obviously not perfect, but I feel like I know more about what to look for specific things and how to use background as a tool to set things up instead of just like having it there for like whatever reason.
Filler. Okay. What about the rest of you?
I think it's helped me interpret data a lot better. Because like when we first like got all this data, I had no idea like how I was gonna look at it and how I was gonna put it into a graph. But now, you know, looking at it, I'm like, okay, yeah.
Okay, good.
I think this entire process has given me a lot more confidence when it comes to doing research, because I'm like, oh, I actually can do that. Because when I walked into the lab, I was like, I can't do this. But now you just did it.
You just did it. And we told you that you would be able to do it when you got here. You just didn't believe us. Any other reflections?
I think the lab helped with our confidence a lot, but even the first 2 weeks, whenever you sat us in a room and asked us questions and you wouldn't give us the answer, like you made us think for ourselves, that really helped my thinking process. I think will benefit me in the future for sure.
Okay. Most students find that infuriating, by the way. But it's a tried and true method that I have. It's one of my pedagogical techniques, is to ask you questions and then just let these long silences hang until somebody just finally gets so uncomfortable with it that they give me an answer, whether it's right or not. Connor is laughing because he remembers those times.
Oh yeah, and every time I answered, they were wrong.
Those times, I don't know what you guys are talking about.
What's that?
I'm still doing that, filling up the awkward silences.
So Connor brings up a point. You know, he's like, yeah, and every time I answered, I was wrong. Do you have a different relationship with being wrong than you did when you walked into class?
It's not as embarrassing anymore.
So you felt
Rather-
embarrassed at times?
Sorry.
Oh yeah.
So is there ever any really negative consequence to being wrong?
No, when you're wrong, you usually find out the right answer. So
Yeah. So one of the things it's not ever been one of my like kind of stated goals in teaching, but one of the things about science is you spend most of your time being wrong about things. You have a particular hypothesis about what the relationship is going to be between seed size and seed number, and the thing that you thought was going to turn out that way turned out not to be that way. And it's not that you made a mistake, that's just the way the data turned out. So I hope that one of the things that you guys have learned this semester is that it's okay to be wrong. It's being wrong is a natural process, a natural part of the process of learning things. And the only time that there's really any negative consequence for being wrong is on an exam. But hopefully you've been wrong enough before you get to the exam that there's less being wrong on exams than otherwise would happen. So, you know, as you move forward into your later classes, don't be afraid to participate because you think you're going to be wrong. Being wrong is actually part of the process. It gives us something to build on, actually. Other reflections?
A nice way to think about being wrong is if you're right about everything, then there's nothing left to know.
Oh my gosh, I don't have anything to teach you. So yes, that's very true. Anything you want me and Dr. Judd to know about possibly doing this again with a group of students next year? For example, I know we could be a little more on top of things in terms of getting protocols out and stuff like that. That was because we were generating all this stuff from new. Hopefully next year's class won't be quite as slow as this one was in terms of the timeline of things. But that's largely because we've done it once now. What other things can we maybe do that differently that might help improve this kind of experience?
I feel like it might be helpful to have maybe more discussions about like why you chose to write the protocol the way that you did and like what the actual steps were for, just to make sure that everything, like every step has a meaning.
Okay, that's good advice. Yeah, sure. Hold on just a minute. Let me write that down. I've written part of it down. Yeah, there's actually a lot in that response I want to get on. Okay, cool. What else?
One thing that I was thinking of was just like ensuring more like communication between the two different lab days, because I know we had issues with that.
Yeah, okay. Yeah, one of the things that we could have done, hold on, I'm writing. Yeah, one of the things that we probably should have done and we didn't do, and I'm not sure why we didn't do it, except we just didn't think about it. We should have given the two groups in the two different lab days, we should have given you like who was in which group and which lab. And then you guys could have done more communication across labs. And we just didn't think that that would be as important as probably it was. But yeah, that's a good idea also. Other things you can think of.
This is probably more specific to the life history group, but I know the reason that we made our mistake of gathering the wrong species was because in the protocol it said look for the white fluffy inflorescence. And so we picked the whitest and fluffiest we could find, and it ended up being a different species.
Yeah, and it was probably a species that bloomed earlier in the year and as a result matured earlier in the year. And so when you went out and found it, you found more of that than you found of the one that we were actually asking you to. So yeah, and that also gets back to Alexis's comment about, you know, being more clear about what it is we're asking you to do and why we're asking you to do it that way and things like that. That would definitely help. Next year, I'm probably going to be on sabbatical during the fall, so Dr. Judd is going to have to do all of this on his own, which really sucks for him. And so chances are, whether we're in coronavirus lockdown or not, we're probably going to repeat this next year. So seriously, if you have suggestions on how we can do this better, do it more streamlined, et cetera, definitely get your suggestions. Even if you think of them midway through the summer, drop us an email because with both of us working on this, it's a little easier. With a single person working on this, it's going to be even harder. And so whatever we can do for Dr. Judd in the fall of next year, assuming that I'm granted sabbatical, is going to really help him out. So if you can think of things that might be useful for him, certainly let us know, because I think this is probably just for a labor management standpoint going to be the way lab gets done next year. as well. So we're asking for your feedback, not just to fill time. We actually do want to hear from you about these things, because they'll help us with the next iteration of this. All right, then. Good job. I was very pleased today with all of the presentations. Congratulations on overcoming the challenges of working in this environment. You have 3 things to do between now and next week. First thing you need to do is you need to finish writing your paper that you've hopefully already started on. Second thing you need to do is this whole thing has been recorded. I'm going to post today, this afternoon, there is a self-evaluation where I want each of you to go back and view your presentation and do an evaluation of how you did. And there's a form that we have for it. Fill out that form and submit it to me via email. And well, actually, I'll put a link up actually and submit it to the Moodle site. And so do that self-evaluation by Monday.